Bio 181, Chapter 14 on DNA. And we'll talk a little bit about DNA maintenance. So, DNA is found in units called chromosomes, linear units in eukaryotic cells. And again, these are made up of a sequence of DNA and some proteins, histone proteins. But at the end, there's a tail region. So telo means tail, like in telophase for the end of mitosis and meiosis. So the tail end of the chromosome is called a telomere. And it's just the region at the end. That in stem cells, those are cells that not differentiated. So these are cells that can turn into any type of cell. So they have multiple potential. They could be a muscle cell, or they could be a nerve cell, or they could be an epithelial skin cell. It could be a bone cell. So they've got lots of options, and those are found in embryonic cells, right? Or sometimes in it turns out that this end actually shrinks. So every time it's replicated, the chromosome gets a little bit shorter. I'm going to exaggerate this. So here's the chromosome. Okay. But at some point, um, it's going to be shrunk so that it starts to delete DNA that's used to code for proteins. So the end is a region that has some extra base pairs on there that don't usually code for anything. But at some point, you're going to get into a region of DNA that's going to be actively coding for proteins. And if you replicate and lose that, then the cell is no longer functional. So this has been a key aspect of research to look at limits to cell life, like how cells age and why cells don't live forever. So it's an enzyme that's found in stem cells called telomerase, and it's only found in the cells that can turn into other cells. That enzyme comes in and it can extend the region. So it adds on base pairs, here, and DNA, some of those non-coding regions. And so it prevents that ultimate demise of the cell. So essentially the cell is infinite longevity. Adult cells, so cells that have differentiated into muscle or nerve or skin, those cells lack this telomerase activity. They don't have it present, and so they do have a finite lifespan. Okay. So DNA mutation is any change in the original DNA sequence. So M sequence A, B, C, D, C. And then something happens. You say you're replicating it, and you want to make an exact copy, but maybe we make a mistake. And so instead of the nucleotides with cytosine, maybe it's replaced with guanine. Right? So that change is called a mutation. Any difference from the original sequence is called a mutation. So mutations are generally, they generally don't have any effect just because we have a lot of base pairs in our DNA that don't actively code for genes. So silent, if they are in an actively coded area, then they generally have a negative. So they're often going to cause proteins that are made reading this information to have a different information difference in the shape or conformation will cause it to either completely not work or so the mechanisms that will go in terms of how to fix these mutations, there's just a few categories: mismatch repair, and excision repair. I'm asking you to be responsible for in this course. So mismatch repair, this happens quite a bit, and it's easy for enzymes to catch. DNA polymerase has the ability for some for this. So if we have this as a template strand, you would create complementary bases. I mean, go to thymine, thymine go to adenine. And say you made a mistake here, or the enzymes made a mistake, and they, instead of adenine, maybe they put guanine. So these were fine. So this is a mismatch. So there's an enzyme that comes along and it looks for anytime we have an inappropriate base pair, it's actually going to cause a little bit of a deformation in the shape. So the enzyme is going to come along, it will chop it out, and then it will replace it with the correct base. So pretty well researched. This is a type of pair when cells are exposed to ultraviolet radiation, so energy radiation. It often happens that two thymines that are adjacent together. So it will create enzymes that will go in specifically and look for these thymine dimers and cut them out. So again, another enzyme comes in, looks for the thymine dimers, removes them, and replaces them with this is a general type of repair. You have a sequence of DNA, you have a mutation in it, and a general excision repair. That comes in, takes this, moves it, cuts it out, okay, and then adds in a sequence of DNA that is correct, right? So it adds in the non-mutated or the original sequence of DNA. That's it.